I'm excited today because chapter 30 launching into today is my favorite chapter. I think, like in the whole intro bio textbook, chapter 37 is, I think, the best chapter. There's a lot of really cool biology in it, a lot of interesting examples of how organisms sense and respond to their environment, and how internal signals, hormones, control processes that are going on inside the plant. So there's a lot of cool stuff in chapter 37. Now, definitely, before you start studying chapter 37, the objectives, because we're actually going to back to chapter 37 3 different times in all 3 of the remaining plant lectures. And so for each one, we just assigned a certain section of chapter 37. So look at the objectives before going on. Oh, wait, I forgot. I have one other announcement. This is going to be a little cryptic. I apologize. But for those of you who were in office hours on Monday, when everybody was packing up to take off, somebody accidentally took off with something that did not belong to them. And so if you could check your stuff, if you have something that doesn't belong to you, and you could bring it back to me, that I'm sure it was totally, as we were all packed. Those of you who are at office hours on Monday, please check that out and get in touch with me if you have something that is yours. So, so again, we're going to revisit different sections in chapter 37 to as we go through the remainder of the plant stuff. So just check the objectives first before you really delve into reading and studying. Now, another thing I'm going to do here with chapter 37, which does talk about the plant hormones, so they're really interesting. You know, you're going to rather than use this time to sort of catalog, okay, here's this hormone, and here's what it does, and this hormone, and here's what it does. That's stuff that you can read about largely independently. What I'm going to do is do sort of a deep dive into one important aspect of plant response to the environment and one important hormone, really kind of go into a lot of depth, more depth, in fact, that the book goes into on this, and then let you read about some of the other interesting plant hormones that are out there. So again, use the objectives as your guide for that. All right. So what we're going to think about is the phenomenon of photo, which is shown here. These, you know, this idea that plants are going to bend towards the light. So we're going to think about how they do that. And oh yeah, by the way, also we're going to use this as an example to introduce what is probably like a, you know, a top 10 concept in biology, which is signal transduction. You know, organisms have to be able to sense their environment and then respond to a cellular level. And signal transduction is the term that we use to explain how that works. And you're going to see examples of signal transduction throughout every lecture here, remainder of the quarter. And it's again, it's a top 10 concept in biology. So we're really going to think about that. And we're going to use this idea of phototropism, right? The plants sense and respond to light. And so here we have a really simple drawing of a stem, right? We're just simplifying it down to 2 columns of cells. You know that it's way more complex than that, but we're going to simplify it down. These are all living cells. You know, we could draw some, you know, cytoplasm in here to remind us these cells are all alive and doing all the things that living cells do. And now if light comes in from the side here, these are representing wavelengths of light. And I'll just say right now that it's not any wavelength that does this. This is specifically blue wavelengths are the ones that are important for these phototropic responses. Then the plant is going to bend towards the light. And to get bending, what has to happen is that the different halves, the different sides of the stem have to grow at different rates, right? If we look at this cell, these 2 cells here, on the light side and the dark side, right? This one is a little bit shorter than this one here, right? So we get, what we have is differential elongation of these cells. Notice we are not changing the number of cells, right? That happens, new cells appear up in the meristem. Here we're looking at cells that are in the stem, and what we're going to do is change their rate of elongation, right? So just to think mechanically about how this could work, here are some questions. So you can answer multiple questions on this one. Here we go. So it's up on Learning Analytics right now. So read through these. Feel free to talk to people around you. This one is also not graded, but do your best to answer it correctly. So you've got a couple different possible explanations here. So think about which one, and what I would think to recommend, I expect to see a lot of people here making your little stems and thinking about, okay, how is it going to work mechanically if these things start bending. So think these all through and pick the ones that you think are possible explanations. For people that just came in, session number is 8, 3, 3, 8, 5, 6, 8, 6, 3, 8, 3, 3, 8, 5, 6, 6, 3. That's our. Oh wait, is it still? Sorry, I think it's still delivering question 1. Sorry about that. Let me. Okay. All right. So now only one of, well, as you're answering this here, I'm just giving a chance for more people to log on, get answers to this one. So you have a few more seconds. As you're doing this, you can think like, okay, only one of these mechanisms is correct, but it will help us to think of ways that this can happen. All right, so 5, 4, 3, 2, 1. Okay, good. Now we've got that kind of fast because we have everybody in here. So most people went with the first two, A and B, which are both possible explanations of this, right? We could either promote elongation on the dark side of the stem, those cells are going to elongate faster, or we could prevent elongation on the light side of the stem. And again, that would cause this differential elongation that would lead to bending. So it turns out, I mean, you know, again, you wouldn't know this based on any information you have so far. It turns out it's this first one that's correct, that the dark side of the stem elongates faster. And one thing that's kind of nice at the start of chapter 37 is it starts off with some, while you have learning catalytics on, let's do this first. It starts off with some nice classic experiments. So, you know, you've heard of Charles Darwin, and of course he's famous for his theory of natural selection. But the second half of his career was a big focus of his was on plants and how they grow and respond to the environment. And so these are some of the books that he published, many with his son Francis, that were looking at plant growth and development. And so this is a figure from your book, and this is one of his experiments here. So let me go on to this. While you're looking at this, I'm going to give you some time to answer this question here as a on learning catalytics. So let's deliver this one here. Okay, so we're looking at this young shoot growing here, and of course it's in the reverse control. You know, you illuminate it from the side and it shows response. And then they do a series of experiments with it. So you can remove the tip of the plant. How does that change things? We could cover up the plant, so with some opaque material that's not receiving the light. How does that change things? We can also cover the base of the plant there, where the bending is actually taking place. Remember, so at the tip of the plant, that's your apical meristem, where cell division is happening, but any changes in elongation are happening down lower in the stem. So go ahead and just sort of take a look through this, kind of internalize this, and answer the true-false question based on your answer. Does anybody have any questions about what you're seeing up here that I can answer for you? Feel free to talk to other people around you if you want to. When I say action here, I'm talking about the actual bending of the tissue, right? The differential elongation of those cells. Okay, so let's go ahead and get your answers logged in here. Yeah, we're doing. Okay, good. We have almost everybody here, and so you know, not 100%, but most people have gone the best answer here, which is A. This is true, right? That if we take the tip off or we cover up the tip, then the plant is it's not sensing the light anymore, right? So these first two experiments here tell us that the tip is where we are detecting light. But it's these cells here in the stem where we get the response, right? So, and notice that even if they are covered up, even if they are in darkness, right? If we darken that lower part of the stem and prevent it from sensing light, it still is going to bend. So it tells us that the site of perception of light is independent of the site of action. And what this implies, right, is that there must be some sort of communication, right? There's got to be a signal here between these, signaling. And this is a chemical signal, so a hormone, which we'll give a name to in a little while, is communicating between the place where the light is sensed and the cells that actually change the way they're growing to produce the bending. Okay. So I skipped a worksheet a second ago that I'm going to go back to now. Hold on, let me manage this. Okay, good. Good. You can put away learning analytics for now, and we'll open it up later and have some more questions at the latter part of the lecture. Okay, going back, we'll think about this hormone in a second, but I wanted to go back to this. Now we're going to revisit this image a couple of times during the lecture because this is where we're really thinking about, how we get from point A to point B, right? So blue light is going to come into the plant, and then eventually the plant's going to bend towards light. We're going to sort of break down some of the individual steps. And the first step is really the detection of that light. And that requires a protein called a photoreceptor. And in this case, it's called PHOT1 for phototropin. It's an abbreviation of the word phototropin, but PHOT1 is the protein. And it becomes active when it absorbs a wavelength of light. So PHOT1 is activated. Now, we're going to do some drawings underneath this here. So let's just think about what that's going to look like. So PHOT1 is a protein, and so it's got a certain protein conformation. Here's our PHOT1 protein. And like a lot of proteins, it can go into a different conformation. And so I'm going to draw this intentionally, kind of looking so we can see that, it has unchanged, right? And so this is our PHOT1 protein that's now active. So this is a theme you'll see a lot, right? A lot of proteins, especially receptors, they have an active state and an inactive state. And here's the inactive state. And what causes the transition is the absorption of blue wavelengths of light, so that the energy from those blue wavelengths is actually by this protein, is this conformational change. So blue light or blue wavelengths of light, right, initiate this change. Now it can also change back, right? So it can go back to its initial inactive state if that light stimulation is removed. But while it's active, what this does is it has an enzymatic activity. It is a kinase. That's familiar from Bio 161. Kinases are enzymes that phosphorylate other targets. They phosphorylate other proteins or other things inside the cell. And you might remember from Bio 161 that adding a phosphate group to a protein or something like that is a way to really change its shape and change its activity. So this is a kinase that is going to change the activity of many different proteins inside the cell. So this is how we're taking, an environmental signal, energy in the form of light, and converting it into a chemical change inside the cell, right? So the light comes in, this protein senses it, it becomes active, and then it starts phosphorylating proteins throughout the cell, and that changes activity. Okay, so that's kind of the first step is perceiving the light, and we'll talk about some of the subsequent steps here in a second. Any questions though before we on from that? Okay, then to get back to this idea here, right, that there's early experiment. Oh yeah, question, go ahead. You have those backwards. So PHOT1 is a kinase, right? That is its enzymatic activity is kinase, right? So it's going to add phosphate groups to a bunch of different targets. What activates it is the light. Good question, though. Good. All right, so Remember, so this simple experiment by Darwin and his son shows that actually is chemical, some sort of a signal that's being transferred between the tip of the plant and the cells that are elongating. And that is by definition a hormone. So here we have our, you have to fill this out. You've got room for this on page 27 there of the workbook. But these are the characteristics of a hormone. And I'm going to ask Dr. Taylor if she's going to sign off on these as working definitions for both plants and animals. The hormones, they diffuse or circulate throughout the body. So they're produced in one tissue, and it triggers a response in a distinct tissue. So it's going to be made in one cell or tissue, and it's going to travel to another cell or tissue and have an effect there. And the other thing about hormones is they tend to be active at very low concentrations. These are very potent molecules. The one thing that is not listed on here is what type of molecule it is, and that's because hormones are very diverse. You're going to see hormones in this class that are small metabolic molecules, and you're going to see hormones that are large proteins. You're going to see hormones that are hydrophobic, the hormones that are hydrophilic. You're going to see lots of different examples. So chemically, they're very diverse, but these are the characteristics they have in common. And now don't worry about the structure. You don't need to know the structure yet here, but Oxen is the name of the hormone that is responsible for this phototropic response. And so Oxen is the protein we're going to care about. I do think it's worth looking at the structure and seeing, okay, it's a pretty, like compared to say a sugar molecule or something like that, it's pretty big. It's got nitrogen and some oxygens there, so it's going to have some polarity to it. And those might be useful for us to think about later as we think about how this molecule is going to move around through the plant. Okay, thank you everybody. I see people still writing, so I'm going to give you a moment to get the definition down here. Again, do you need to know the structure of Oxen? No, I'm just thought it'd be useful to have up here for later. So now, Darwin's experiment here didn't show that there was a hormone called Oxen that did this. They just knew that there was some sort of signal. So there were subsequent experiments, and you can see this one in your book, this is figure 7.6, where people started to characterize this hormone chemically. And so here what they did is, it's kind of cute, like basically you can follow the red color to show you where the Oxen is. And they did an experiment kind of like what you're going to do in lab starting next week, which is to use some sort of like a gel-like material that has some hormones in it, and you can use this to manipulate growth of plants. So this is sort of a gel here that they use, and you can see that the Oxen diffuses from the tip of the plants where it's made. So Oxen diffuses into this gel. And then it's kind of nice. Then the researchers, what they have is this little gel that is loaded up with this hormone molecule, whatever it is, and then they can start to play with it. So you can see if you put, so here if you apply this gel asymmetrically to the stem, right, we're going to get a higher Oxen concentration over here. So here's where you're going to deliver that Oxen, high Oxen concentration. And that results in the bending. So you can kind of control which way the plant bends by where you apply this Oxen. So it shows that, hey, even without the stimulation of light, by manipulating spatially where the Oxen is, you can control the growth of the stem. All right, so again, you can look at some of these classic experiments here, but they get us to the point of understanding, all right, there is a little chemical molecule that is going to produce these changes. Okay, so now let's get back to this one here. So we've already sort of drawn the first step in this, right? So we're back on this illustration on page 26. So the first step in this pathway is that we get the activation of FOT1. And what happens next is that the Oxen that's produced in the tip of the plant is going to get redistributed. So it's going to get transported, and this is actively transported to the dark side of the stem. So we get Oxen transport to the dark side of the stem. So just to think about that, again, really simple drawing of the tip of the plant up here, right? So there's the apical meristem, and we're going to kind of just divide it down the middle. So we have the the dark side over here, what's going to happen is that auxin molecules, if the plant is illuminated equally from all directions, then auxin is going to be equally distributed through this whole tissue. But when you get this unequal illumination, then the auxin starts to get transported over here to this side. So we're going to end up with a high concentration of auxin here on the dark side of the stem. And this is active transport. So that's why I showed you the image of the auxin molecule before. That molecule is not something that's going to be able to just cross membranes by itself. It's not going to diffuse across membranes. And so there are auxin transporters, pumps and channels that are going to move auxin from inside of one cell out into the environment and into the next cell, and sort of it leapfrogs in these cells to accumulate on the dark side of the stem. So that's the next step that happens, all right? So the light comes in, that turns on this, it's detected by the receptor phototropin that, causes a bunch of things to happen, but one of the things that it happens is the transport of auxin, and so we get a higher concentration on the dark side. So what is that? Oh yeah, question. Yeah, the auxin, I didn't say this, but we'll get into it later. Auxin is produced in the shoot apical meristem, and then it tends to move down the plant. But here what's happening is that instead of moving down equally on all sides, it's being disproportionately moved over to the dark side. Yeah.
What is the activity of the photo-1 protein have to do with auxin?
Right. So you're asking about the connection here, right? So how does I can only answer this in a simple way because the whole mechanism isn't known, right? But there are pumps and channels that move auxin, and most of the time they are inactive, right? But when this phototropin has wavelengths of blue light, it is going to activate those pumps and channels nearby through phosphorylation. That causes them to start doing their job, just to move the auxin around. So that's the fact. Now, we don't know all of the links in this pathway yet. I'll show you some images about that later. But yeah, okay. So here, just check your understanding. Let's think about this. Here we've got, I like showing a lot of mutants where, some protein in this pathway is missing. So here's wild type plants, and you can see they're little seedlings, they've grown up and they've bent over towards the light there. And here's a mutant, and it is, it has a loss of auxin transport. So one of those pumps or channels that transports auxin is no longer working. So we can't do that redistribution of auxin like we see there. So what I get to do is just, picture in your mind or pencil it in there, something. What would you predict about this mutant compared to the wild type? So wild type, we have a normal phototropic response. What do you think the mutant is going to look like in this case if we can't redistribute the auxin? All right. You want to go on record and show your prediction to somebody sitting next to you? Go for it. But, all right. So here it is, right? So here's the mutant. And so, by the way, if we had a mutation in the Photo-1 receptor, right, the photoreceptor, we would expect the same thing. Basically, these plants, they can sense light, they have the receptor, but they can't respond to it because they're not redistributing the auxin, so they just continue growing straight up and down. So anyway, this is something you're going to see a lot here, where I'm going to show you mutants, where one of these genes that we're talking about is mutated so that its protein isn't functioning correctly, and ask you to make predictions about how that's going to change the growth and development. All right. So now we need to think about elongate, right? Ultimately, this is going to produce differential elongation in the cells. And so how does auxin affect cell elongation? That's the next thing we need to think about. And to think about this, I just want to remind you from something earlier in the class. This is the drawing we did for Turgor pressure. Now remember, all these cells, they are, think of them as like they're under more pressure than the tires of a car, right? There is the cell wall that is holding this volume together, and the water that is wants to move into those cells is pushing out against the cell wall, and that creates Turgor pressure. And remember, we can get to this point where the cell is not going to change its shape because the wall is kind of pushing an equal and opposite pressure as the water pushes. So to get that cell to change its shape, what needs to happen is we need to change the cell wall somehow, right? We need to alter the cell wall so that it can, that trigger pressure that's pushing out can cause the cell to expand or elongate. So we have to change the cell wall a little bit to make this happen. That means we have to go into a little bit of a deeper dive into the cell wall, which previously we just said, oh, it's cellulose. And that's true. But now we're going to look in detail here at the cell wall. And you've got this illustration. You have actually ahead on page 29. So we're going to add some stuff to this drawing here. So the primary component of the cell wall is definitely cellulose, but it is in the form of these large kind of like cables, multiple strands of cellulose all kind of woven together. And these are called cellulose microfibrils. Basically a microfiber. I mean, but these are like little cables of cellulose. Yeah, I'm sorry, I should orient here. This is the plasma membrane right here, PM for plasma membrane. This is the cytoplasm. And so then this is the wall out here. We're looking at sort of a higher resolution image of the wall than we thought of before. So you've got these cellulose microfibrils. I just threw this in just because it's kind of interesting how this works. You know, these cellulose microfibrils are made by proteins in the membrane called cellulose synthases. Cellulose synthase, which takes individual glucose molecules inside the cell here. So these are, you know, glucose and it's beta glucose, you know, in one of the conformations of glucose. So beta, they go into these and out come strands of cellulose, and these kind of get woven together here. And this is a multi-subunit protein, so it's actually going to create like multiple strands of cellulose at once. And so this cellulose comes out of the cellulose synthase and essentially gets sort of woven together into these rather complex microfibrils, we call them. Okay. Now, if we stop our drawing here, you'd be like, okay, well, that's one image of the cell wall, but it seems like, you know, if you got pressure pushing out, there's no reason why these microfibrils wouldn't just slide past each other and allow the cell to expand. Because what we also have is another kind of molecule that is important for, we would say, cross-linking these together. And that's a little molecule called hemicellulose. So it's a modified form of cellulose, hemicellulose. And I'm going to draw it as this little wiggly line here. But what it does is it essentially like forms cross links between these microfibrils. So, you know, there's going to be all these little hemicellulose molecules sort of binding these together here. And it's this combination, right, of these big cable microfibrils and then these little connections called hemicellulose that give the cell wall an important quality of being like, you know, slightly flexible, but only within to a certain extent. Now, the hemicellulose is manufactured inside the cell. Here's a little vesicle down here. It's manufactured inside the cell, and then it is released into the wall through exocytosis, and then it binds onto these cellulose microfibrils. All right. Now we're going to come back and add some more stuff to this drawing in a second, but this is the basic, or this is a slightly more nuanced vision of the cell wall. So again, we want these cells to be able to expand, and to do that, we have to find some way to sort of loosen up this structure to allow that expansion to take place. So that's what we're going to think about next. Now, rather than tell you how that works, I'm going to ask you to look at, oh yeah, here's just a nice textbook image of this where you know, you can see that one of these microfibrils here is actually made up of many, many strands of cellulose, all kind of woven together. And if we magnify it enough, then we get down to individual glucose monomers that make those up. Okay. So we're going to look at some experimental results. This leads into a learning catalytics question, but maybe we have time to look at that. But if you want to start getting learning catalytics, if you want to log back in again, you may. I'll go ahead and start it right now so we can have this. But you're going to think about it first before you enter any answers. Okay, so we're going to think about ways we can cause the cell wall to loosen up. And so here's some experimental data we're going to look at. And first show you the setup. All right, so here's a live plant, a little seedling here. And we're going to cut off this little tiny bit of stem. So it doesn't include the apical meristem. It's just a little chunk of stem. Then we are going to very gently kill all the cells in that little piece of stem, right? Because we're going to get rid of living cells, and we just want the cell walls to be maintained. And so, so we're going to do this by freezing the tissue that kills the cells and then thawing it out in a gentle way that lets us just preserve the cell wall. So basically, these are just cell wall only. And then, and this is a very precise little machine that you have to design for this, we're going to suspend that little piece of stem. So here's the little chunk of stem that we have. Again, it's just the cell walls now. There's no living cells there. We're going to suspend it, and we're going to hang a little weight off the bottom, right? And so we're going to see how stretchy is this little piece of cell wall. Okay? So we've got our cell hanging there in this little solution, and then there's weight down here at the bottom. It's going to be pulling on it. We're going to ask, okay, can we do anything? Can we modify? Oh yeah, and then so the stem is hanging in this little bucket of solution here, and we can modify that solution. We can add things to it. We can change different chemical properties about it, and then we can ask, all right, to change the stretchability, the flexibility of the cells. Okay, so that's the setup. Anybody have any questions just about the experimental setup before we start looking at the data? Yeah. Okay. So then down here we have different trials, basically all set up the same way. And the first thing I want to say is that, like, if we look at number one, it's pretty easy here, right? We're going to hang that little piece of stem there for about an hour and a half. And in this case, we don't see any change. It doesn't start stretching. This one is done entirely at pH 7. And unless it's stated otherwise, all of these are done at pH 7, unless the pH is changed. All right. So basically that's saying, all right, we keep the solution at pH 7 the whole time. There's no change. We look at number 2 here. Okay, here it's at pH 7 for the first 30 minutes. And then we're going to change the pH. We're going to change it to pH 4 for the rest of the. Now we do see that it's actually starting to lengthen, right? That little bit of stem is lengthening. All right. So you have 6 separate experiments to look at. I'm going to give you some time to look at all of these. Work on them yourself, talk to people around you, whatever you want. But there is a question that is now running where you're going to select all of the true statements on here based on the have. All right. So I'm going to give you some time. I'm going to come around and answer questions if anybody has them. But so you have a few minutes to look at all of this data and answering these questions up here. So. I'm going to go back to the data slide because you can see all of these statements up on Learning Catalytics, so you shouldn't need to see them while you're working on that here. All right. So go ahead, take some time, think about it. I'll come around, ask you if you have any questions, and we'll think about what chemical, how can we modify the environment to allow the cell wall to change its shape. Okay. Go ahead and work on this. There's a lot to take in, so just kind of go question one graph at a time and see what little bit of information that gives you. I would say, thinking back to those of you who took my 161, thinking back to one of the labs in my 61 is probably going to be helpful, really hands-on lab. So week 2, and let me know if you have any questions. So people are asking about, I guess, 3, 4, and 6. You know, maybe I could give a little more nuance to this as you're working on it. I said extract from expanding. So maybe we could say like, okay, young cells early on in the stem, you know, that are in that process of changing their shape and differentiating versus, you know, mature cells, right? So these are older cells that are younger, you know, they have basically reached their sort of functional maturity, whether that means they're alive or dead at functional maturity. They're not changing their shape anymore, right? So anyway, I don't know, does that help clarify that a little bit? Right, okay. Let's see how we're doing here. All right. Who's active has answered here. So I'll give you a few more seconds just to get those answers in. So remember, the whole point of doing this is to ask, like, okay, what sort of conditions in the environment can loosen up that cell wall and allow a shape change to take place, right? That's what we're doing here. All right. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Okay, good. Looks like we got pretty much everybody here. So selecting the true statements from this, and by the way, you can close down learning catalytics right now if you want to, once we're done with that. So the true statements, and almost everybody had this, is the first one and the last one. Those are our two. We have ways that we can eliminate the others in every case here. Oops, hold on just a second. Oh yeah. So cell wall expands if the environment becomes more acidic. That's shown here, right, by an experiment number 2, where decrease the pH, right, make the environment more acidic. That does allow the cells to become here, right? So yeah, we know this from, you know, experiment 2. It's going to give us that information. All plant cells contain a substance that promotes cell wall expansion. Well, that's where we have to consider, like, you know, down here, if we, especially comparing, you know, 3 and 6 to each other, right? If we use, you know, young cells that are in the process of changing their shape and differentiating, then an extract from those cells does have this effect. And whereas if we do mature cells that have already stopped that process, whatever this substance is, they don't seem to have it anymore, right? It doesn't have the same effect. So that tells us that one would be false. So experiment 3 and 6 let us see that. All right, maximum cell wall expansion is seen when an extract from an expanding cell is applied and the pH is kept constant. Now, you know, this is the one here, experiment number 4, we see the biggest change. And there we're changing both the pH and also adding that extract. So that's where we get like the biggest effect. Oh, yeah. Okay.
Number 2, it's not just about like the essence of the transported protein rather than
You know, I think maybe this is just based on the data tables that we have. You're asking about mechanisms. We don't really know. We'll get to mechanisms later, but this is just saying like, oh, you know, some cells, you know, you grind up these mature cells, they don't have any effect. If you grind up young cells that are in the process of changing their shape, there is something in those cells that does loosen up the cell. And somebody, what evidence that a protein is involved here? For the last one, yeah, go ahead. Let's say, okay, so that's true and not the answer that I was thinking of, but you're maybe even thinking about like pepsin and pepsinogen, right? That you learned about in the last lecture, where there's an enzyme that only becomes active in pH. That actually, so you are completely right. That is 100% true for this protein. But how do we know, yeah, that it is a protein involved?
Experiment number 5 was one of it,
Right, yeah, so that boiling stuff, right? If we, whatever that thing that was coming out of these cells that caused the cell to expand, it's killed by boiling, and that is at least good indication that there's a protein involved, because treatment would denature the protein. Nice job, good. So, all right, so what we know is that pH like lowering the pH lets the cell wall expand more, and then also there's some protein that is found in some types of cells that will also cause the cell wall to expand. So let's go back here and we can draw a little bit more into this diagram here. So first of all, let's think about the protein. So the protein is called expansin. I'm going to use a E with a circle around it to represent this. And so expansin is also, along with our hemicellulose, it gets secreted into the cell wall. And so it's going to hang out in the cell wall. And again, like we said earlier, similar to the hexinogen protein that you heard about in the last lecture, it is activated by a decrease in pH. And what does it do when it's active? What it does is it attacks and degrades the hemicellulose, those little connections between the microfibrils. The expansin is going to cut that. So right, it's going to cut these hemicellulose bonds here. So the expansin, I should add this in here. So expansin, it cuts hemicellulose. And, oh, sorry, it's going to kind of low. It is active at low pH. So in acidic environments, it becomes active. Sorry, that got kind of small down there, right? So expansin, it cuts the hemicellulose strand, and it's active at low pH. So that combination, right, this extract from growing cells and the low pH really allows the maximum sort of stretchability of the cell wall because we're activating this expansive protein. Okay. Yes, question. You know, I put them together in the same vesicles. They both get secreted out into the cell wall, right? And the expansin might be hanging out there, and then if the pH drops, it's going to suddenly become active and start breaking up the hemicellulose, right? Yeah.
So is the reason why having like extract from another plant cell and then a low pH increase the rate more expansion in the wall?
So the thing here is that those younger cells that are actively changing their shape, they are making more of this expansive protein, right? And secreting it out into their environment. Okay, now the thing we still need to do here is tie this into Oxen, right? We started talking about light and Oxen, and now we're talking about the cell wall and making it more stretchy and everything. Now we're going to connect those two things, right? The Oxen picture and the wall picture. And we're going to do that. I'll jump all the way over to here. Using the, well, I guess, okay. This picture sort of helps us bridge the gaps in your worksheets up on page 31, right? And so here again, we have little pieces of stem that are floating in water. And if we add some Oxen to that, if you look carefully at these, you can see that they start to elongate. And these aren't dead pieces of stem. These are little live pieces of stem here. So this is just making a connection to the idea that Oxen is going to promote elongation in these stem tissues, right? So now we have to think about how that happens, right? How does the presence of Oxen cause the cell walls to eventually loosen up so that the cells can expand? All right. So to think through this, we're going to do, these are the two most important drawings of this whole lecture, and two things that I want you to really think about a lot and make sure you understand. A lot of this is also covered in the text, but I'm going to add some extra stuff as well. So on page 30, you've got two identical drawings here. And in the first one, we're going to think about when, it says no Oxen present. I'm going to change this to say very low Oxen levels. Probably it's wrong to say there's never any Oxen around. So low Oxen. And then in the other one, we're going to have high levels of Oxen. Okay. Here we're going to draw some things into this cell here. There's the plasma membrane, we have the nuclear membrane, the endoplasmic reticulum membrane inside here and everything too. So we're going to start off by drawing one thing that we need, which is an Oxen channel. It's going to allow Oxen to enter the cell. So Oxen is one hormone that actually enters the cytoplasm of cells. You're going to see some hormones that never do that. They always stay outside of the cell and they bind to receptors on the surface of the cell. But this is one that goes into the cytoplasm. And inside the cytoplasm, there are two Oxen receptors. And one here is just sort of hanging out in the cytoplasm. This is called the Oxen response factor, ARF. So Oxen response factor. This is a protein, and so ARF is for Oxen response factor. And it is hanging out there just. The other Oxen receptor is here in the reticulum membrane. It's called the Oxen binding protein, ABP. So Oxen binding protein, ABP. So these are two proteins which can bind to Oxen, and if they do, it's going to change their activity. There's one other protein we need to draw in here because this Oxen binding protein, it's in the endoplasmic reticulum, but it is never by itself. It is always co-localizing with another protein. And this is something you're familiar with. This is a proton. So it's a proton, an ATP, a proton pumping ATPase, right? It's going to use ATP energy to generate a proton gradient. But right now it's just sitting there in the endoplasmic reticulum. And so now this drawing is done. Basically, this is the cell that is waiting to respond to Oxen, but there's very low oxygen in the environment, and so we don't have much going on here, okay? But that's so this is sort of our baseline, okay? Now we're gonna draw this thing to think about how all the ways that this is gonna change when Oxen concentrations go up, all right? So let's switch over to that one here. So here's our Oxen channel again in the membrane, and now let's draw a lot of Oxen here in the environment. So we're gonna have a bunch of Oxen molecules, and some of those are going to find their way into the cytoplasm. All right. Now, I thought use a feature here on my iPad. Like you might don't draw for a second, just watch. So here we have the Oxen response factor here in the cytoplasm. It is going to bind to Oxen. So let's show a little Oxen molecule bound onto the side. Now here's where I'm going to be cutie with this. So when that is bound, this Oxen response factor undergoes a change in its conformation, which allows it to actually be imported here into the nucleus. And it is a transcription factor. So it's going to leave the cytoplasm and it's going to go into the nucleus and it's going to bind onto the DNA. So that's what you should draw in there. But I kind of wanted to show the cheapo animation as it's happening, right? So the Oxen response factor is going to move, it's going to move into the nucleus, bind to DNA, and then it's going to gene expression. So it's going to change gene expression, or I'll just write gene expression down here. And actually, there's a bunch of genes that get turned on when Oxen is present. One of them, this might not surprise you to hear, one of them is the expansin gene. So we start making more of that expansin protein when the Oxen arrives. So gene expression, so one example here of a gene that's induced by Oxen, one example is the expansin. Right, so we're going to get an increased expression in expansin to make more of that protein around. Okay. So that's one change that happens when Oxen. Now, what about we have another Oxen receptor that was there in the endoplasmic reticulum. Now I'm going to, again, maybe don't draw what I'm about to draw here because I'm going to do my little animation thing again. So here's our Oxen binding protein, ABP. And remember, it is co-localized here in the endoplasmic reticulum with this proton pump. And so again, an Oxen molecule is going to bind to this. So here's our little Oxen binding on there. And then that causes the whole thing to move from the endoplasmic reticulum up to the plasma membrane. So that's what you should draw there is the ending state, right? So this is going to move, and it moves in a vesicle, right? That's how things get from the endoplasmic reticulum to the plasma membrane. They move, a vesicle leaves the endoplasmic reticulum and takes this up to the plasma membrane. It gets there, then we start pumping protons. So remember, acidifying the environment, right, is one of the things that causes the cell wall to loosen up, right? So now this oxen binding protein moves up to the plasma membrane. It brings this proton pump with it, and then that starts to pump protons out into the environment, right? So that's going to lower the pH of that extracellular environment. So really good, really important sort of before, after kind of pictures here. And you should be able to explain the different proteins, what they're doing, and how this leads to this change in stretchability of the cell, right? This change in the cell. So just to review this again, we have these two, when the oxygen concentrations are low, we have the oxygen response factor and the ABP ready to respond. And then when oxygen arrives, a bunch of stuff is going to start changing, right? Gene expression is going to change, and that's catalyzed by the oxygen response factor. And then the pH outside the cell is going to change, and that is at least partly involved with oxen binding protein. All right, any questions I can answer about this part here? Okay. So really important that you understand these diagrams well and can really explain a lot of the stuff that's shown on here. Yeah.
Why is that ABP plus compound always
So the question is about this mobility from the endoplasmic reticulum. So every proteins that need to end up in membranes or in the nucleus or something like that, they have essentially little ID cards, right? And those ID cards are in the form of small amino acid sequences that are part of the protein. So what you can imagine, maybe it's easier to think about oxygen response factor, right? So it's got this little signal that's called a nuclear localization signal, right? Which says, hey, this is a protein which needs to go to the nucleus. Without oxen, that part of the protein is kind of curled up, right? Think about protein conformation. And when the oxen binds to it, it opens up, and then that nuclear localization signal is detected and allows it to move into the nucleus, right? They don't know where they're supposed to go or anything. They're just moving in response to chemical processes inside the cell. And and there's proteins that are destined for the plasma membrane that have special amino acid tags on them that say, hey, this is a protein that needs to undergo, that needs to move from here to the Golgi apparatus and then up to the plasma membrane. So it's all based on sequences embedded in these proteins, which are either hidden or exposed when they bind to oxen. Yeah.
What signals the ABP to move that?
In both cases, these are receptors, right? We label these both as oxen receptors, meaning they're both proteins that bind to oxen, and that changes their activity. Yeah. I don't think it's right to say oxen is transporting them. Are you thinking back to earlier slides where we're talking about oxen transport? We'll do the whole picture now. Yeah, hold on. Because that's the next step here, right? So we get back to this now. This is the big picture thing. So to rewind this all the way back to the beginning. Blue light hits the plant. That causes the Photo-1 receptor to become active. That causes oxen to move from the light side of the stem to the dark side of the stem. So we create this high concentration of oxen on the dark side of the stem. And then that causes the acidification of the cell wall in those regions, right? So the cell wall acidifies. And this is, again, only on the dark side because that's where those oxen concentrations are the highest. And that leads to this last step, right? So if those on the cell walls are looser and more stretchy on the dark side, and that's going to let those cells expand more quickly, and that produces the bending that we see, right? We started off by thinking about, oh, what would have to happen for that bending to take place? Yeah.
So for the new plants where they're oxygen transporter,
Mm-hmm.
it wasn't working.
Right.
Was there a life cycle
what was happening was that the cells were still expanding, but they were all expanding at the same rate, right? By having this high auxin on one side and low auxin on the other side, we create a case where they're going to expand at different rates, and that causes it to bend. So if you don't get auxin, like a plant that's illuminated equally from all directions, it's going to have equal auxin concentrations in all of the cells. So they're all going to sort of expand at the same rate. So this is the uneven light is that redistribution. Yeah.
Where does the Oxen binding factor find the Oxen? Why is it? I'm sorry.
The auxin binding protein. Sorry, you're asking where?
Yeah.
So the auxin would, but I'm just going to draw on this here and then erase it, right? So it would bind to the auxin when it's there on the endoplasmic reticulum, and then it causes that to move up to the plasma membrane. It moves to the plasma membrane when it's bound to auxin. And this is good to talk about this. You're going to see lots of examples of receptors, right? This whole, the next, 5 or 6, almost every remaining lecture in the course is going to have some sort of thing like this, where there's a chemical signal, it binds to a receptor, and that changes something that cell is doing, right? Gene expression or some other thing. Yeah.
You said the receptors are being like vesicles.
Well, this would be a vesicle would be the way that you would get from the endoplasmic reticulum up to the plasma membrane. Again, like this would probably be better to show as an animation rather than these two static drawings. Yeah. Right, we could add that here, like when I say decrease the pH, all those things are going to happen. The expansion gets activated, it cuts up the hemicellular. So all that stuff from the previous slide is happening as a result of this. That's right. Yeah. Okay. So remember, I started off by talking about signal transduction. So this is a good example of a signal transduction mechanism, right? How the arrival of a molecule, auxin in this case, totally changes a whole bunch of stuff happening inside the cell. It changes gene expression. It changes the location of proteins inside the cell. So this is a good example of gene expression. I just want to point out chapter 37 starts with sort of an overview of signal transduction. And a lot of students look at this figure and be like, oh, that's the auxin thing we drew. Be careful. This image at the start of chapter 37 is not the auxin pathway. It's just sort of a generic example of all the sorts of things that can happen in a signal transduction pathway like this. So it's really useful to look at. But keep in mind, this is a little bit, this has some things in common with the auxin pathway we just drew, and some things that are different. All right. I think I'll go to this. All right, so let's imagine here another mutant. So in this case, you've got, that's on page 31. Here we have a wild type plant. And now for this, let's assume this one is being illuminated equally from all directions. So it's not getting, don't worry about bending in this. We're just going to think about the elongation of the cells. What would you predict about a plant that is missing the Ox binding proteins? Or if it doesn't, there's a mutation that for making that receptor. Go ahead and just think about what the mutant plant would look like here. Think about draw a little sketch of it. We're basically thinking about how to compare to that wild type if we took away the ability to respond to auxin. Question? Yeah. So the question is expansion responsible for all of the elongation in the plant? I think that, yeah, so expanding cells need to loosen up the cell wall, right? And so expansion is the primary way to do it. I guess something I didn't list on the slide too. When you decrease the pH in the cell wall, it actually has another thing, which is it does slightly loosen the connections of the hemicellulose to those big microfibrils. So even without the expanses, the lower the pH still does make it a little stretchier, but the expansion is what you need to really have a cell that's going to elongate rapidly. All right, so hopefully everybody has a prediction here. So here's what the mutant is going to look like. So basically, it's still going to make leaves and flowers and all the parts, but they just don't expand, right? So they're, without being able to sense and respond to auxin, we have very, very limited expansion of the cells. So we're making all, like these two plants, they probably have the same number of cells in them, right? It's just that in the plant, there's been a lot of expansion of those cells. And so the auxin binding protein is going to severely limit that. All right, here's another, now, so that was phototropism. Let's change our attention just for a second to another tropism, another plant movement, which is gravitropism, right? And so here this little plant is bending not in response to light, but in response to gravity. And this is also the same mechanism that we talked about for Oxen is present in this. Now, one thing that's different here is how gravity is sensed, right? For phototropism, we have a light receptor, phototropin 1, that initiates the whole thing. With this, there's a different receptor that is sensitive. The plant has a way of sensing gravity. I'll let you read about that in the textbook, and there's objectives about it too. But the plant is detecting gravity and changing the elongation, the rate at which the cells are elongating. So here, we're not going to do this with learning catalytics, but we can just do top side of the stem, low side of the stem. So we've got this, this plant bending up. So on which side of the stem do you think oxygen concentrations are going to be higher? Is it going to be on the top side of the stem or on the low side of the stem? Where would it produce bending like this, right? Okay, so supposed to be going thumbs down here, which is correct. We're going to get higher oxygen concentration on the low side, right? So and that's going to cause those cells to start to elongate faster, and that causes the plant to reorient itself toward it. Now, roots do the same thing. Interestingly, here the pathway is, and I'm sorry about this, it's quite different in terms of how the roots respond to gravity compared to how the cells respond to gravity. But roots will also reorient themselves. So we can look at this really simple drawing of a plant laid on its side here. What's going to happen is that we're going to get, just to zoom in here, we're going to have high oxen on the low part of the stem, and that's going to cause rapid elongation. Actually, I won't use the term rapid. I'm going to say that will promote elongation, to use some vocabulary from earlier. So promote elongation. Now, what's going to happen in the root is actually the same thing in terms of oxen. The oxen is going to get redistributed down to the low part of the root. And so we're going to end up with a high concentration of oxen down here. And here, I'm just going to introduce you to a concept that you're going to see a bunch of times also in the cell, in the rest of the class. Different cells in different parts of the plant body respond differently to the same hormone. And if you're a root cell, high concentration of oxen inhibits elongation. So it's just exactly the opposite of what happens in the stem. Here, this is going to inhibit elongation. So same hormone, but a totally different response because we're in a different type of cell. So inhibit elongation. And the result of that is that, the root cells are going to bend down, right? Because the cells on the top are going to be elongating a little bit faster. Whereas here in the shoot, right, the cells on the bottom are going to be elongating faster, which causes the shoot to bend up like this, right? So I include this just to point out that, hey, you're not going to be able to say, just because this hormone has a particular effect in this type of cell, that it's going to have exactly the same effect in other types of cells as well. And this is a good example where the roots respond in exactly the opposite way to the cells in the shoot. Yeah.
Is oxen produced like throughout the whole entire plant, or is it shoot?
So where is oxen produced? Actually, that leads into our slide very nicely here. So oxen, well, our next two slides. So I'm going to now summarize some other things that oxen does. And so as we just talked about here, and I will get to answer your question. So oxen is involved in photo and gravitropic responses. So responding to light and gravity, gravitropisms, phototropism, and gravitropism. Some other things that oxen does. One of the things that's involved in the phenomena called apical dominance, which is the way like the shoot apical meristem actually communicates with all of those axillary meristems that are involved in producing branches. And oxen is sort of the hormone that's used for that. So apical dominance, and I'm not going to say much about this because this is going to be something you'll start in lab next week. There's a really nice experiment that you're going to do with apical dominance. And the other important thing that oxen does is it promotes cell division in roots. So basically, it tells the plant to make more roots. So promotes cell division in roots. Okay. Now, before I move on, now we're going to start talking about a different hormone. So there's another hormone called cytokinin, and cytokinin sort of has the opposite profile from auxin in that it promotes cell division in the shoot apical meristem. So, and I have another figure that's going to show this a little bit nicer in a second, but cytokinins promote cell division in the shoot. So basically we have one hormone whose one of its roles is to say, make more roots. And then we have this other hormone whose role it is to say, make more leaves, make more, photosynthetic parts of the plant, right? So, and there's an importance talk here between these two hormones, auxin and cytokinin. And we can summarize this here. So auxin, and here's to answer your question, auxin is made in the shoot apical meristem. That's where most auxin is synthesized. And then it moves down the plant. If the plant is illuminated equally, it's going to move equally down the stem, and it affects those cells as it goes down. If the plant's illuminated from the side, then some cells are going to get more auxin than others. But the auxin moves down the stem and eventually gets into the roots, and it promotes cell division in the root. So it's going to make more roots. This other hormone, cytokinin, has the opposite profile, where it is made in the root apical meristem, and it promotes cell division in the shoots. So these are the hormones that the root and the shoot use to talk to each other. And I think if we draw up, we could do some simple drawings over here to help see why this is important, right? So here's a simple plant. We've got a shoot system, and it's going to have a root system. And now, if we have those two systems have to grow in proportion with each other, right? Because the shoot system is going to provide the photosynthates, the sugars that the root need for metabolism. The root is going to be absorbing water, nutrients that the shoot is going to need, and so they're going to grow in proportion to each other, right? This plant here is with a tiny little shoot system and a huge root system is going to have a problem, right? There's not going to be enough photosynthesis to support the growth of the roots. On the other hand, if we have a giant shoot system and a teeny little root system, then we have the opposite problem, right? Where now structurally this plant isn't going to be very stable, and there's probably not enough surface area down there in the soil to absorb the water and nutrients that are needed. So I think it kind of makes sense that we need coordination between the shoot and the root, and they have to sort of grow in proportion with each other. And it is these two hormones, auxin and cytokinin, that coordinate that growth between them to ensure that both of them are sort of growing proportionally. So we've got one signal, auxin, that's going from the shoot down to the root. We have another hormone, cytokinin, that is communicating from the root up to the shoot. All right, so I think that's where we're going to have to stop for now. There's a few more important things. I'm probably going to need to make about a 5-minute video follow-up for this just to get to the last couple of points. But remember, there's a whole lot of other plant hormones. You're going to read about gibberellic acid and ethylene and abscisic acid. They're all really cool and have interesting roles in the plant. Refer to your objectives to learn a little bit more about those. And of course, come to office hours, and I'm happy to talk about it. But my goal for today was to go really in depth into one aspect of signal transduction, so you can see how this hormone, all the cellular events that allow it to regulate plant growth. All right, I have office hours starting in about 10 minutes over in my office, so you both come by. Oh, sorry. Dr. Taylor's at 10:30 also, so both of us are available this morning. Okay. Can you again with an email, and I'll go look at it now. I think that's what we said. Oh yeah, I got it. Let me go now.